There's different kinds of selection. Not all of it has to do with survival, but some does. So if we look at life stages, we'll start with a zygote. That means the egg has been fertilized, and and will start dividing, and you'll get the embryo. Now, if there's a mutation, that's a bad mutation with the zygote, it will become unviable. In other words, it's got a bad mutation, and it may develop up to a certain point, but that's about the end of it, and then it's dead. Now, once the organism actually develops into an adult that then can produce gametes, either a male or a female, then the adults actually have to find a mate. In many organisms, like plants, they send out pollen, or they get animals to do it. But in animals, there's also something called sexual selection. And sexual selection, we're going to get into it a little bit, but there are different sized gametes between males and females. Females make bigger gametes, eggs, compared to sperm. And that sets up a difference between the sexes. So, after they, or during, after, when they're adults, they also have to survive selection. Now, when they produce offspring, or they produce seeds, you have fecundity selection, and that's the number of babies or offspring they produce. Like some plants produce a lot of offspring, and there's variation between some individuals and others. So we'll look at that too. And then there's gametic selection. So it turns out gametes can actually compete for fertilizing each other. And then sometimes genomes are not compatible, obviously if they're different species, but also even if they are the same species. And so we'll talk a little bit about that as well. The bottom line is some phenotypes leave more offspring than others. So this is selection on survival. They like the green ones. The orange ones are what's left over. Rabbits. Australia was overrun with bunnies because they were brought by European settlers, English. There were no real predators that were killing them, because we're dealing mostly with marsupials, and all the marsupial predators had been wiped out by that time. And basically, they had a rabbit explosion. Ate everything. No food for the sheep, no food for the kangaroos, nothing. Well, they found this virus in South America that the rabbits there have. And they found out that it worked on these other rabbits to the point where it killed about 99.98% of the rabbits. It was it's called myxomatosis. You can see these different rabbit plagues along different years. And around here is when they started using it, and it killed everything. Now, that's selection. That's like Ebola virus, right? It turns out, though, some rabbits were more resistant to it than others, and so over time, some rabbits were able to survive. But there's still, even the rabbits that actually survived, there was still about 30% mortality instead of 99.8%. And the virus itself evolved. Why? Well, it was in a different environment in terms of what rabbits had what alleles that made them resistant. So there was ongoing selection pressure that's called coevolution. Okay. Fecundity selection. So a plant with a lot of fruit and seeds versus one that doesn't produce as many. Now there's something called sexual selection. It's usually males are competing against each other for access to females. That's why females in our society are treated like a resource that males want to control. Now, it also turns out because males and females both provide resources for raising offspring, the same thing is going on. There's female-female competition for males as well. And that's why okay, so these guys are just fighting it out. This is the male, bird of paradise. Here's the female. And the male has these showy plumes. The one that's most dramatically pretty, that's the one the female generally chooses. And then, of course, for us, mate choice. Now, the reason there's sexual selection is this. Oh, we'll get to it. Okay. So, there's different kinds of selection. There's directional selection. I'll show you examples of each. Stabilizing selection, disruptive selection, and then negative frequency dependence, but you don't really have to remember that. This is a diagram of different types of selection. So we have, up above, we have this population, and the phenotypic measurement can be, let's say, coat length or behavioral phenotypes and anything like that. And then on the y-axis we have the number of individuals. So if we look at A, on the left, we have stabilizing selection. That means individuals that are closer to the mean have higher fitness. So as you can see, there's going to be selection against the biggest and the smallest. So they all die. They all die. And you're only left with, finally, a much narrower number of individuals in terms of, let's say, size. We're going to use what's called body birth weight and stabilizing selection for that. Directional selection, get into that. That means there's a trait. Let's say you have denser fur. So we'll say less dense there, 20 there, centimeters. And in this case, if the climate gets colder, all these individuals here will be wiped out. And eventually, you're going to end up with a mean that it started here, but now it's over here. And then we have disruptive selection. This is where there's selection on the average size, let's say. They all die. They start dying here, of course. Dying here. And eventually, we'll have two different means, a smaller and a larger. Let's say you have a distribution like this. So we'll say 0 to 20. And this is, we'll say coat length. So it'll 0 to 20 centimeters. And then here is the proportion or the number of individuals who have, let's say, fur that is this length. And we'll say that looks like that's 10. Now, if there's directional selection for shorter fur, because it gets warmer, all these individuals with long fur get wiped out. And this is all that's left. And remember the old, that used to be the mean, the average. And now what do we have? Over time, we'll end up with a population like this, where the mean now is 4 instead of 10. That's directional selection. That means you're selecting for one form versus another, and it's changing, it's selecting for traits that are, let's say, bigger or smaller, something like that. You can also have, just so you can think about it, there's the same thing, where there's selection for bigger, right? Again, so now we have, there we go. Now we have a population. Instead of, let's say, 10, now we're at 18. That's directional selection too. So here's some example of directional selection in beak length in the Galapagos finches, the Darwin finches found in the Galapagos that Darwin collected. A whole bunch of different species. There was a drought. So prior to the drought, this is population size. Let me actually do it on here. Okay. So this is population size, and this is time. The drought started right around here. It reduced seed availability, and it turns out, by the way, there's variation in beak size, and beaks were highly heritable. And by the time we get to here, the only seeds left over, there aren't very many, but the only ones left over are really, really hard seeds that only the biggest beaks can actually crack. In fact, if you look at right here, this is now the population size. It's like down around 200 instead of up here around 1,400. In other words, almost everyone died. Only those with the strongest beaks survived. It turns out most of those were males, by the way, but a few females did. So, here we are, the distribution of beak size along the bottom, and the number of finches. And before the drought, the mean was right around, let's see, what was it? About 8. After the drought, where the population went down, the population mean was now about 9.8. Don't do that. Cancel. Okay. So that's the response to selection. And we can also show how heritable it is and how things change, in that if you plot the parent's bill size versus the offspring's bill size, you can actually see the response to selection, that over time it goes bigger and bigger and is highly heritable. So that's directional selection. There's another kinds of selection. Okay, this and a good example of stabilizing selection is birth weight in humans. Obviously, babies have big heads. Small babies don't survive as well as large babies. They pass finely through the birth canal with no problem. Unfortunately, large babies sometimes don't. And not only do they not survive, but the mother doesn't survive. So here, this is the optimum birth weight is now here instead of over here. So, what did I just do with that? Oh, here it is. Okay. So, in this case, all these individuals over here are selected against. Small babies don't survive as well. Large babies end up not making it through the birthing process. So this is called stabilizing. Around the optimal size is around there, and it doesn't. The mean doesn't really change all that much, the average, but the variation around the mean does. There's much less of it. It's called stabilizing selection. Individuals at either tail have lower fitness. Here we go. Have lower fitness than the mid size. And here we'll say it's whatever size it is, we'll say 10. Okay? Now, if the population, if we plot this again, what we'll have is a population like this. Everything will be clustered around the average, and individuals that are too small don't survive as well. Individuals that are too large don't survive as well. With stabilizing selection, the variance around the average decreases, but the average actually usually doesn't really change all that much. So like with directional selection, the average size, let's say, does change from smaller to larger, larger to smaller. Stabilizing, if the average was, let's say, 10, whatever it was, the average will still probably be 10, but the actual variance around the average will be much less. In other words, small individuals and large individuals will not be around anymore. Okay, now we have something called disruptive selection. Okay. we have the same sort of situation. Now, disruptive selection means that individuals that are around the average are disfavored or selected against. Oh, come on. Okay, so, why isn't it doing it? Okay, so you can understand. So now we actually have two different means or averages, a small size and a large size. Individuals that are in the middle don't do so well. This is disruptive selection. Here's a finch. It eats seeds. The thing is, there's all different kinds of seeds. There's small seeds and there's large seeds. And the size of the bill determines the efficiency of actually eating the seeds. So in this case, birds, and there's really competition, let's say for medium-sized seeds, they all disappear. So you've got two different seed populations, and now you've got birds that are being selected either for lower, smaller width or larger width. In other words, the mean, all that was here, has been wiped out, and now you've got two different modes, or averages. A small mandible width that goes with small seeds, a large mandible width that goes with big seeds. Everybody getting this? Okay, if you have any questions, let me know, because we're going fast. So I'll give you a little view of negative frequency dependent selection. These are salmon. Anybody in here ever fish for salmon? Sorry, asked a bunch. Okay. Anyway, this is on the Kenai Peninsula up in Alaska. I think that guy goes about 97 pounds or something. He's turned, so he's probably not very sweet looking, sweet eating either. And then on the left of these little salmon, same species. They're both males. If you think the jack, how do you think that the small one will do against the big one in terms of competition for a female? Huh? Get his ass kicked, right? Nothing's going to work for him. Now, the big one, it takes 4 years from the time the eggs have laid, spends about a year in the natal stream and goes out to sea for the rest of the time and then comes back to spawn. That's 4 years of being exposed to predators. The small one, it only takes 2 years. It comes back to the natal stream every 2 years. Much smaller, right? And you think, well, so what? It's still small. You know, bigger is better. Well, it turns out it's a sneaker. Because when a female, what she does is she digs out a red, an area, and lays her eggs. And then the male comes along and releases his semen, fertilizes the eggs. Well, if you were underwater, you'd see there's all these different sized rocks, some farther, some nearer to where she actually makes the red. And behind those rocks, the little guys can, you know, no one's seeing them, right? They're hiding. So while two big males come along, each one trying to fertilize the eggs, this little guy is coming in and under the radar fertilizing the eggs. Now, it turns out that there aren't very many places to hide. So usually individuals of small size, they have the highest fitness when there's very few of them, because otherwise they'd be competing for rock sites. The same thing goes for the big ones. If they're rare, they don't have to fight each other. So then they're favored, but those are things that are going to change over time in terms of the proportion of each, or number of individuals of each type. So as the frequency of, or proportion of one group goes up, their fitness goes down, and the other, the smaller size, let's say, goes up, and the other way around, the big one goes down, because there's too many of them, they become rare, goes up. Yeah. Did you, okay. So, this is a kind of thing, now these are two different kinds of males. In this case, of course, bigger is not better necessarily. Depends on the environment. There are actually probably, there's probably a third kind of male here. And if it's not here, there's other examples. And this is a male that is in drag. Looks exactly like a female. Everybody treats him as a female. Big males don't want to, they want to mate with him, but they won't chase him away. So he hangs out near another big female and is hiding in plain sight and actually fertilizes the eggs. You guys got to go salmon fishing at least once. Anybody ever been fishing? Oh, good. Oh, boy, that's good. Okay. How about hunting? Excellent. Finally, good. I'm at least a couple of them. Hunters are. It seems like there's strong negative selection for hunters, doesn't it? It's screwed up. Okay. Oh, there's one other thing. Remember when I said that individuals in the medium size, they're not selected against, they're selected against? Well, this also is actually an example of disruptive selection. Again, you're either small, able to hide, or really big, able to battle. Okay, so sexual selection. It depends not on the struggle for survival, but on how well individuals are at getting mates. And there's, if they're battling together, the two males, it's usually based on size, who's bigger and who's stronger. But it can also be what are called sexual characteristics, like the plumes here, that the female decides on who to have sex with. Now, this is called an Irish elk, and if we look at it, this is the size of the man. It was huge. They obviously fought. Why does sexual selection exist, and what does it have to do with Arnold Schwarzenegger? And a male toadfish. So what does Arnold Schwarzenegger and a male toadfish have in common? They both produce small, cheap sperm. There is something called it's anisogamy. What it is different sized gametes. Males generally produce lots of small, cheap gametes, and females produce fewer, larger gametes, the eggs, that have more resources in them. Because of this asymmetry or difference between gamete size, it creates an automatic conflict between the strategies of the sexes. The female is providing these expensive eggs. The male is providing these really cheap sperm. Female reproductive success is usually limited by the resources that she invests in the eggs. She only has a limited number of resources. Males provides lots of resources for his sperm, but he ends up with tons and tons and tons of sperm. So a male's reproductive success, unlike a female's, is limited by the number of females he can mate with. Now, before you guys all get uppity about, oh, this is why men cheat and women are pure and like pure maidens, it doesn't quite work this way, as you probably know. But in theory, there's other factors that go into it. Now, it turns out females have a limited reproductive success because they have a limited number of eggs they can produce. And so, what's called the variance, or how many females make this many eggs, how many females make 1,000 eggs, how many females make 10 eggs, well, they're usually all around for the same species around the same amount, because there's an upper limit that they can make. Males, on the other hand, they make sperm, lots and lots of them. The thing is, for most species like that elk, most of the males have almost zero reproductive success. Because when they're fighting, they always lose. The ones that win have a big harem of 40 or 50 females. He mates with all of them. So the male, in this case, can have many, many offspring compared to the female. But most of the males they're pretty unhappy. Okay. Human sexual selection and biparental care. Now, remember, there's intrasexual competition. That means within the same sex for mates. So, we'll say intrasexual, so males compete against males, and females also, for humans, compete against females. There's also mate choice. Who are you going to choose? So that's between a male and a female, both ways. And then there's a multitude of mating systems that humans actually have. There's also something called sexual dimorphism. That means males and female humans look different and act different. Continuous female receptivity and concealed ovulation. Well, it means that normally in the animal world, there's only certain times, or any other plant or anything, there's only certain times of the year that individuals breed. The rest of the time, they're not interested. Well, with humans they are, but males can't tell when a female in her cycle is the most fertile. Now, there's some thought that maybe they can, with a sense of smell, but they're not aware of it. Of course, females have sexual selected characters that males find generally nice. Breasts and buttocks, cute, right? Okay. They have this nice waist to hip ratio. They have minimal body hair, or less than males. And think about their voice. It's not some low voice. It's usually, it's sonorous. Women have beautiful voices. Okay? Just lovely. Now that's a sexually selected trait as well. Now, males, they don't conceal ovulation. They have concealed feelings. They never show their feelings. They have a V-shaped, this is Conan the Barbarian, muscle definition, a low, deep voice, much grunting. Apparently, women like a low, deep voice for the most part. I don't know. When I hear a low, deep voice and there's a lot of grunting, I usually run away. And then they're hairy and aggressive. So, the reason, though, is for both females and males to compete against other females and males against other males, and they're both making choices, is that for the most part, males provide a lot of parental care, paternal care. So he has a small sperm, but he's sticking around to help raise the offspring. Whether it's just providing, safety or resources in the form of food. And so, like a lot of birds, songbirds that are monogamous, serial monogamy, where it takes two to actually successfully raise an offspring. Humans have the same thing. But humans have a lot of other mating systems as well. Anybody hear about harems, that males have harems of women? Yes. The Sultan of Brunei? Right. Okay. Having many wives. Well, that's polygynous. That's a polyamorous or mating system. So not all humans are just monogamous. There's even a few cases where resources are so there's so few that there's one female and a couple of males in Nepal, in the high Himalayas. Now, it's actually dying out at this point because people are moving into the cities. But in the past, really, usually it was two brothers. They were related to each other. They had one wife. And it took both of them so that they could successfully raise offspring. And then there's something else called, now, this is not a value system. It's a sexual mating system. It's called promiscuous mating system, where males and females go out and have sex with many different partners. Okay? That's also found in nature. All these kinds of different mating systems are found in nature. Humans do most of them, or a lot of them. Though compared to other animals, our mating systems are pretty tame. Oh, too bad. Okay. So first I want to talk about, this is the reading frame. It turns out, if you look at each one of these, now those are not nucleotides in DNA. We have C and G and A and T, okay? But it takes 3 nucleotides in a row to code for what's called an amino acid. And there are 20 different amino acids that we have. And amino acids make up proteins. So DNA, all it's doing is you're just transcribing it from DNA into a protein. So there's a reading frame means that it's called a codon, and you need 3 letters of the DNA code, C, G, T, A, in each space, and that codes for a specific amino acid, and then you put all those together, you get a protein. Okay. Now, sometimes there are mutations in the reading frame. So this is what it normally spells. Dad pet the fat dog, the fat cat ate the big rat, the guy saw the U and the dough. Okay. The reading frame has now switched. Either there's been an insertion that moved everything down one from that point on, or there's been a deletion. Okay? And so the reading frame is now basically making gibberish. And usually if a gene, there's a mutation that for a gene that actually makes a protein, it's usually deleterious. It means it's bad. Okay? There's a lot of, it's easier to screw up a protein because it can't work anymore than it is to get something that actually works better. This is two spaces. It also ends up with gibberish. In other words, so these are the two that have been dropped. So the reading frame is now DPE, as you can see, DPE, DPE. So remember, these are codons. And the codon is made of 3 base pairs of DNA. Now these are words filling in for the DNA, but the DNA are either C, T, A, or G. Okay. Okay, in this case, we're doing a deletion in the middle. We're actually deleting an R right here. So we're not going to have a rat anymore. In fact, from the cat ate the big, from then on, it's all going to be gibberish again. So if you shift the reading frame 1 or 2, you're then out of the reading frame, and you're going to get gibberish. So the R has been deleted, and now we have Dad pet the fat dog, the fat cat ate the big heg ooze. Now we're going to actually do a deletion of 3. A codon is made of 3 base pairs. And so we're going to change the meaning, but we're going to still be in the reading frame. So dropping this, and what we get is the IGR has been deleted, and now we have ate the bat, the guys saw the U and the dough. Okay, so we've got a different meaning, primarily bat instead of rat. So it depends on this amino acid, how much it changes the actual protein. It may be almost as good, or it still may be completely deleterious. Okay, now I just want to say, mutation by itself is a very, very weak force, because mutations don't happen very often. Plus, we have a mechanism for DNA repair. So it's being as DNA is being transcribed into what's called RNA, single strand. If there's a mistake, there's all these different biochemicals that go along and actually fix them. So in general, mutations are really rare. They may happen, but they're fixed. So it's by itself, as being random and very slow, it's a very weak force of differences in individuals. Okay, very weak. Of course, if you put it together with natural selection, then becomes very strong. But both of them do. But you need to have both for natural selection to actually occur. But mutation by itself has very little effect. A lot of the mutations have no effect at all. Okay. Now, there's also something called genetic drift. So you have random mutations that show up in the population. If you have a very small population size, so let's say we're going to flip a coin 10 times. And let's say we flip it, obviously the probabilities are 50% heads, 50% tails, but with only 10 individuals, it varies by 10%, so you sometimes can do 10 flips and you'll get 6 heads and 4 tails, or you could even, with very low probability, get 10 heads and no tails. Okay? So if you have a population of only 10, you can change the frequency of certain alleles much more easily in terms of the percentage of alleles in the population. If you do the same thing and you, let's say, have a population of 1,000, so you have 1,000 flips. Now, probabilities are the same, 500 and 500, but instead of 1 out of every 10 in that change, you have 1 out of 1,000. So the percentage doesn't really change very much. If there's, let's say, 540 heads and 460 tails, well, that's a lot of mutations, but it's not nearly, that's like 46% versus 54%, right? Instead of 50% or 60%. So this coin flipping actually goes on every time people make gametes. Because when a parent creates a gamete, they either take all the chromosomes from one parent or the other. It's like a 50% split. So you get either the chromosomes from your mother, and you get the chromosomes from your father. You don't get both. There's some crossing over, and shuffling, but that's generally what happens. So the chance that's strictly a chance thing, like flipping a coin about which alleles you get. You get the alleles from your mother or your father. So this is what genetic drift is. It's random. And alleles change in frequency just by random chance alone. And they are likely to change much more rapidly if it's a small population than a big population. Now, you could also think about it this way. This is something that would not, this is small population, and this guy steps on randomly, let's say a couple of green ones. There's not selection. It's selection happening, but there's never going to be evolution by natural selection. The bugs aren't seeing birds. This is some random guy steps on some bugs and kills them. That's a random sort of thing. But most of the genetic drift comes from this, getting half your DNA from your mother or your father. So this gives you an example of drift. On the top, we have a population of size 4. That's a very, very, very small population. The chances of an allele going either to fixation up here, that means you have a couple of different populations, and this allele, just by drift, random chance, ends up the whole population goes to A1, or just by random chance, this allele goes extinct and it's replaced by this. But you can see how wide that is and how quickly it happens. That's because population size 4. The same thing is happening down here, but this is population size 400. And so drift is much weaker in this situation. Eventually, things will probably go to fixation, but it will be, thousands of generations in the future. They just randomly change in terms of frequency. So the frequency is either it's A1 and A2, or big A and little a. Let's say little a is here, big A is here, in terms of the proportion. And the populations vary just by drift. So in a big population, genetic drift has a very weak effect. In fact, if you looked at something like a population size of 1 million, it would be even much less than that. So the takeaway for this is that genetic drift is a force of evolution. It's not an adaptive force. It is strongest in small populations and is weakest in large populations. Okay? That's the bottom line. Okay. You might have heard of population bottlenecks. Now, as I said, drift changes according to population size. So if we reduce the population size because of drift, first of all, it may be just a random, it will still have the same proportion of alleles, but there'll be fewer of them all over completely. Or you can end up with a change in allele frequencies after all these individuals have, let's say, died. So you can lose alleles. And then there's something called homozygosity, where it's the same allele. We get an allele from our mother or allele from our father. It could be the same allele, or the allele we get from our mother is A, and our allele we get from our father is B. Now, in this case, we have this population. We have high mortality. And so all of these go away except for this group right here. And the genotypes, by the way, we haven't got to that, but we will. This is homozygous for allele A1. That means you get an allele from your mother and an allele from your father. If it's the same allele, that's homozygous. Homozygous. If it's different alleles, A1 and then A2, it's called heterozygous. So the top is homozygous for A1, A1, and we're diploid, so we have always two alleles, homozygous for A2, or we have an individual who's A1 and A2. This is called a population bottleneck. The frequency of these different genotypes is much different than the frequency of the original population. That means we've lost a lot of alleles. In fact, there are no more red alleles or orange alleles at all. There was some catastrophe, lowered the population size. That's all that left. So from then on, they may breed successfully and build themselves up again, but the population as a whole has lost genetic diversity. And in fact, they've lost one allele completely. Gesundheit, whatever. Okay, so this is called a population bottleneck. It could radically change allele frequencies just because we're going from a large population where drift is weak to a small population where drift is strong. Founder effect. You have a bunch of individuals in one place, and they disperse, a few of them disperse to another place, and they start a new population, but it's a small population. So we go from a big population with a whole bunch of different alleles to a few individuals going and starting a new population. The subset of individuals probably has a different frequency of alleles, just like we had with population bottleneck before, when the whole population was reduced in size. Which is a bottleneck. Usually you would think it would happen, let's say there's a big disease or something like that. So this is just starting a new population, and it's a small population. So there's this little island called Tristan da Cunha. That's a British island out in the Atlantic Ocean. There were some British colonists that 15 British colonists went there in 1814. And of course, they bred, and they got a bigger population. Unfortunately, one of them had a recessive trait that was deleterious, that was bad. It caused blindness, as a matter of fact. It's called retinitis pigmentosa. So if each colonist had 2 alleles for that locus, there was a total of 30 alleles for the whole population, and one individual was a carrier. He didn't show pigmentosa, retinitis pigmentosa. He was a carrier of it. And then they bred, and they bred, and they bred, and let's say just by chance, because there's no real strong selection until it's 2 of the same bad allele together, that's recessive, until all of a sudden, over time, because it started in the population, there's a high incidence of this autosomal recessive trait. Even though there was only 1 allele in the total population with a total of 30 alleles. Just by drift alone, it increases the population size. It's actually selected against if you get 2 of them together, but it's a real problem there. So it's like 10 times higher in frequency, this disease, this genetic disease, than you would find in a normal population. So that's called the founder effect. You go, individuals from a large population go to a place and start a new population. It's usually a small population. It might get big eventually, but the initial conditions often determine what the population is going to look like in the future.